Hello, my name is Samantha Bentley, and I will be talking about case number 2 and case number 4. So in case number 2, if we are looking at how glucose is taken up in different tissues, we first want to look at whether it's in the fed or the fasted state because the fuels utilized differs between the two. So first we'll look at the fed state where insulin is high. So in the fed state, glucose is the primary fuel used for all the tissues, so the liver, the brain, skeletal muscle, and adipose tissue. They are all oxidizing glucose, and in some cases they're storing it. However, the primary glucose transporter does vary between the tissues in the fed state. So each tissue utilizes a different glucose transporter. So first we'll talk about the liver. And in the liver and the fed state, glucose is taken up in an insulin-independent manner by the GLUT2 transporter, which is always present on the cell membrane, regardless if insulin is present or not, hence the insulin-independent manner. So in the liver, glucose is oxidized through glycolysis, which will eventually enter the TCA cycle and have glucose synthesis, fatty acid synthesis, as well as the urea cycle will be active. So it will also store glucose as glycogen through glycogen synthesis, and it may also enter the pentose phosphate pathway. So in the brain, in the fed state, glucose is also taken up in an insulin-independent manner. However, this is through the GLUT1 and GLUT3 transporters where glycolysis is occurring. On the other hand, both skeletal muscle and adipose tissue take up glucose in an insulin-dependent manner by the GLUT4 transporters. So skeletal muscle will oxidize glucose through glycolysis and will also store glucose as glycogen. At the same time, protein synthesis is occurring as well as the TCA cycle as a means of oxidizing acetyl-CoA in an aerobic manner. Adipose tissue is oxidizing glucose through glycolysis as well, and it stores excess glucose as fatty acids and subsequent TAG synthesis for storage. So that's pretty much the fed state. So now we'll talk about the fasted state. And in the fasted state, the liver's primary role is to provide glucose for both the brain and the red blood cells for glucose homeostasis. And it does this through glycogenolysis. I think I said that wrong. Glycogenolysis, which is the release of stored glucose, and gluconeogenesis, which is the synthesis of glucose. So for its own fuel source, though, the liver utilizes free fatty acids through lipolysis in the adipose tissue. So in the brain, glucose is still taken up in an insulin-independent manner, and it's oxidized through glycolysis. Skeletal muscle in the fasted state is similar to the liver in that it oxidizes free fatty acids through beta oxidation. And then adipose's tissue main role in the fasted state is to provide those free fatty acids through lipolysis, which frees the stored free fatty acids so that they can be oxidized in the liver and skeletal muscle, which I just talked about. So now that we've discussed that, we'll talk about how this is different in cancer cells. So glucose uptake in cancer cells is characterized mainly by a large shift from the usual reliance on oxidative phosphorylation towards glycolysis. So this is more advantageous for cancer cells because they require much more glucose than a normal cell does, and glycolysis is the least energetically demanding process and the most time effective. So there has been differential expression of glucose transporters in cancer cells. Specifically, we're looking at the GLUT1 and the GLUT3 in the glioblastomas. So there has been found to be overexpression of the GLUT1 and GLUT3 transporters in areas that are moderately hypoxic and with the highest amounts of hypoxic-induced factor 1 alpha, or HIF-1 alpha, which is a protein that is largely transcribed in times of hypoxic stress, such as the tumor's inner regions. So the overexpression of the GLUT1 and the GLUT3 occurs to provide energy where oxidative phosphorylation is not as readily available due to the decreased availability of oxygen and or the modulation of the mitochondria by cancerous cells. However, when specifically looking at these brain tumor cells like we are in this example, GLUT3 transporters are more transcriptively active because while GLUT1 is broadly expressed in both the glial and neurons, GLUT3 is restricted to just the neurons. So in order to alter the glucose uptake in cancer cells, the BTICs outcompete for glucose uptake by co-opting the GLUT3 transporters because, like I said, the brain tumor requires a continuous source of energy and molecular resources for new cell production. Okay, so now we'll move to case 4. And, sorry. All right, so answer choice A is not correct. Because the mother states that the baby is only eating the breast milk. So if it's only eating the breast milk since birth, it only contains glucose and galactose. So therefore, the baby is not ingesting fructose, so an intolerance to fructose cannot be causing these symptoms. Answer choice D is not correct because a deficiency in the glucose 6-phosphate dehydrogenase usually doesn't appear in individuals until they're exposed to foods, medications, or infections, which can cause the red blood cells to break down prematurely. So basically, this infant is only exposed to breast milk, so it hasn't really been exposed to anything else. So it's hard to say that these symptoms are caused by this glucose 6-phosphate dehydrogenase. So this early destruction of red blood cells is called hemolysis and leads to symptoms such as shortness in breath, rapid heart rate, and the yellowing of skin color, which doesn't really match up to the symptoms that this infant has, such as the jittery spells, the low glucose, and an enlarged liver. So I'm going to eliminate answer choice D. And then this leaves us with answer choice B and C, which is much more difficult to eliminate because the symptoms of galactosemia and glycogen storage disease are very similar, such as the low blood glucose. So first we'll look at galactosemia, which means galactose in the blood. And when infants have this condition, they're not able to metabolize galactose, which is found primarily in breast milk and cow's milk. So infants with galactosemia typically have symptoms right after they have that breast milk, such as diarrhea or vomiting, which didn't occur in this case, so this is kind of making me question galactosemia. So then I'll move towards the fact that epinephrine was administered and there was no blood glucose increase, but rather an increase in blood lactate. So this is likely to happen with an individual with galactosemia because it affects the breakdown of glycogen, so not really galactose. So that being said, I eliminate galactosemia, and I believe the correct diagnosis is answer choice C, glycogen storage disease, or GSD. So GSD, glycogen storage diseases, are a group of genetic disorders that cause glycogen, which is the storage form of glucose, to be improperly stored in the body. So some of the symptoms that are associated with GSD are the low blood sugar and an enlarged liver due to that buildup of glycogen. So this is kind of on track with what this infant has. In addition, the jittery spells are most likely due to the low blood glucose levels, and the epinephrine that was administered, it causes the liver to convert stored glycogen to glucose and then releases it, which should raise those blood glucose levels. So in this case, since the blood glucose levels did not increase, that's why I'm thinking it's GSD, because of the inability to break down that stored glycogen, which would increase the blood glucose levels. And that increase in the blood lactate could be due to the Cori cycle, which basically when epinephrine is administered, skeletal muscle glycogen is broken down and released as lactate, which then should be converted to glucose in the liver. However, since the infant has GSD, then there is an inability to convert that lactate to glucose, causing an increase in the blood lactate levels. So I believe that the glycogen storage disease is what is causing this infant all of these symptoms.